Welcome to Chapter 17. This chapter is looking at a few complex patterns of inheritance. In Chapter 16, the previous chapter, we talked a lot about really simple patterns of inheritance with a few little variations. These are called Mendelian inheritance. So this is where we have our simple dominant recessive traits where we follow Mendel's two laws, the law of segregation and the law of independent assortment. We also looked at X-linked traits, so what happens when traits are sex-linked or only found on the X chromosome and what happens with that. We also talked about incomplete dominance a little bit, where the heterozygote shows kind of this blending of both of your homozygote traits. We looked at codominance with the blood typing, and then also sex-influenced traits. So sometimes the sex of the individual can influence if a trait is dominant or if it's recessive, like in the male pattern baldness. Then we also introduced you to a little bit on how the environment can influence how traits are actually expressed in an individual. This chapter we're looking at a few more complex inheritance patterns, and we have 7 that we're going to look at. I'm going to go through these pretty quickly, and we're not going to go into a lot of depth with them, but you should at least have a general idea of kind of what this pattern is and how it might influence different inheritance patterns. The first 2 complex inheritance patterns that we're going to look at involve gene interactions. So far we've looked at traits that are just influenced by one gene. So this gene codes for fur color, for example. That fur color can either be black or white. But most traits, like hair color, skin color, they're actually influenced by multiple genes. So we have multiple genes that contribute to that phenotype or to that trait or characteristic that's actually being expressed. So this is a gene interaction. So when we have a single trait, like skin color or fur color or flower color, this one trait, and it's actually controlled by 2 or even more genes. And each of these genes we know has 2 or more alleles. So you can see that we can have a lot more than just 2 alleles in a population. We can have a lot more than just one gene with the 2 alleles that will control this trait that we're looking at. So it can get very, very complex when you start to have this gene interaction that occurs. One type of gene interaction is called epistasis. This is when alleles of one gene mask the expression of alleles of another gene. So it's kind of similar to when you have a dominant or recessive allele, except that your 2 alleles in one gene can be dominant over the 2 alleles of a whole separate gene, and that can influence the trait that is expressed. It can influence that phenotype. So here we have an example of this epistasis that can occur. So when we look at this cross in our P generation, we have 2 white flowers up at the top on the left-hand side. So if you look at those genotypes, when you cross those 2 individuals together, your F1 generation, you're going to be heterozygote for both genes. You have a big C, big P, little p. When you allow that heterozygote to self-fertilize itself, you would expect a 9:3:1 ratio. But instead, we get a different type of ratio. We end up with, there's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 individuals that will end up being purple, and the other 1, 2, 3, 4, 5, 6, 7 individuals will end up being white. So we kind of have this, we don't have this 9:3:1 that we usually expect from a dihybrid heterozygote cross. Instead, we get a 9:7 ratio. And this is because instead of just having one gene that codes for flower color, we have 2 genes that are involved to produce this flower color. And see, our big C is dominant to little c, the big P is dominant to little p, so we can follow Mendel's dominant recessive genetics right now. But then if you look, if you have 2 little c's, it will actually mask the expression of the big P. So if you have 2 little c's, you're going to end up with a white flower, even though you have that dominant P. If you have two little p's, it's going to mask the expression of the big C. So here we're seeing that if you have two recessive alleles in one of your genes, it's going to mask, or it's going to stop the expression of a dominant allele in your second gene. So this is an example of this epistasis. You have one gene will mask an expression or stop the expression of another gene. And then the other gene interactions, so we've been talking about traits. So far we've looked at what are called discrete traits. So discrete traits are where we have really clearly defined phenotypic variants. We have white flowers or purple flowers. We have black fur or white fur. We have tall plants. We have dwarf plants. It's one or the other. But most traits, you don't get one or the other. You get kind of a continuous variation. These are called quantitative traits. And the majority of our traits are actually quantitative. So they show continuous variation over a range of phenotypes. So this would be like if you have a range of flowers from white to dark purple, you could have light purple, medium purple, kind of dark purple, very dark purple. So you have this range over that phenotypic color. And these traits are also polygenic, so they're also controlled by more than two genes, oftentimes 3 or more different genes. All these genes contribute to the outcome of this trait. And then in addition, environment also plays a big role in how these traits are expressed, or at what part of that range of phenotypes you actually will see on that individual. So an example we have here is turkey weight. So there are 3 genes, we'll call them W1, W2, and W3. A big W codes for a heavy allele, so that turkey will have an extra pound. The little w, the recessive trait, codes for a light allele. So if we look at our 3 different turkeys here, we can have little tiny turkeys where their genotype will be all little w's for each of the 3 genes. We can have a medium-sized turkey where half of those alleles are the heavy allele, the big W, the other half are the little w for the light allele. So this will give us a medium-sized turkey because half the alleles are heavy, half are light. To get the heaviest turkey, all of your alleles have to be that dominant allele. So they're all big W's on the right-hand side. Another example is skin color. So you probably noticed that people have different skin colors, and we have this big range of very, very pale skin all the way up to very dark skin. And this is also a continuous trait, and it depends on how many alleles you have for dark skin. So for example, in this chart, in order to get very pale skin, most of the alleles in the individual code for light skin. In order to have very dark skin, all the alleles have to be the dark-skinned allele, and you can have any number of combination. So the more dark-skinned alleles you have, the darker your skin will be. You'll produce more melatonin, which causes darker colored skin. So here we have this continuous variation that can occur with this quantitative trait that's also polygenic, or multiple genes contribute to it. And then in addition, skin color we can also change by different environments. So in the winter, generally people have a lighter skin color compared to in the summer when you're exposed to the UV radiation and you become tan. Then your skin will look darker. So the environment also has a big role in how these genes are actually expressed, or what your phenotype is going to be. Our first 2 complex inheritance patterns, we were looking at different types of gene interactions and how that can change the phenotype that's actually expressed in our individual. So the next one we're going to look at is called gene linkage. This is when genes are found on the same chromosome. So far we have assumed that all of our alleles or genes that we're looking at have been on different chromosomes, and that they follow Mendel's law of segregation and Mendel's law of independent assortment. So this inheritance pattern that we're going to look at, this is when we don't see traits that follow Mendel's law of independent assortment. We actually see traits that are linked to each other because they are found on the same chromosome, or they're found very close to each other on a chromosome. So this is our gene linkage. So gene linkage is when your two genes are close, they're on the same chromosome, they tend to be transmitted as a unit because during sexual reproduction, chromosomes are transmitted as a unit unless you have crossing over that occurs. So crossing over can produce new combinations of genes, and these are called recombinant chromosomes when you have that crossing over that occurs in meiosis. But most of the time, these linked genes are passed down as a unit, so they're passed down together. The parental types, these are offspring that show the same genes as the parental generation, so the offspring's traits do not change from their parental generation. So parental types are when these two genes are linked together on the same chromosome, they get passed down as a unit. However, during meiosis, we have that crossing over. We can also end up recombinant individuals that end up with different combinations of genes that are different from what the parents have. So I mean, this is again how we can get some variation in our offspring. So here we have an example. We have two traits. We're looking at flower color, either purple or red flowers. And then we're also looking at pollen shape. So you either have long pollen or round pollen. So we would expect if these two traits were independent of each other, they followed Mendel's two laws, we'd expect a 9:3:3:1 ratio. And that is shown on the right-hand side. Our expected ratio is 9:3:3:1, because that's what we'd expect when we have a heterozygote dihybrid cross. But when we actually plant these flowers, you do the cross, you plant the flowers, you collect the data, we observe a ratio of 15:1:1:5. So this observed ratio does not match what we expected from this heterozygote cross. This is an example of gene linkage. So we find that purple color and long pollen, those two traits are actually linked to each other. They're passed down together for the most part. The red flowers and the round pollen, they are linked to each other. They're passed down as a unit. And those are those parental offspring. But we get a few individuals that will have different combinations. Those are your recombinant offspring. So this is our gene linkage. So far we have 3 complex inheritance patterns that we've looked at. So we're going to continue to go through the next 4. So the next one, extranuclear inheritance. These are genes that we find in different organelles, not in the nucleus. So if you remember way back when we talked about cell biology, we talked about eukaryotic cells, the DNA is in the nucleus. That DNA condenses down in the chromosomes that have the genes on them. We also talked about 2 organelles called the energy organelles or the semi-autonomous organelles. These are the mitochondria and the chloroplast. In mitochondria and chloroplasts, they actually contain some of their own DNA or some of their own genome. So these chloroplast and mitochondria that contain their own DNA, it can be passed down differently than the DNA that's found in the nucleus. So most organisms, they get their chloroplast or their mitochondria from their mother in the egg cell. This is called maternal inheritance, and it's the most common transmission pattern that we find in mammals or other organisms. So females tend to invest more into their offspring, so they give their offspring the mitochondria and the chloroplast if it's a plant. There's other types of inheritance that you can also see with this extranuclear genes. There's biparental, where both the pollen and the egg contribute chloroplast to the offspring, and generally we only see this in plants. And then even more rare is paternal inheritance, where the pollen contributes the chloroplast and the mitochondria. And really we only see this in pine trees, where the pollen or the male parent contributes more of the organelles. But in general, we see maternal inheritance, so the mom will invest more into the egg, she'll put mitochondria, she'll put chloroplast in there. And then that offspring will get that mitochondria and chloroplast DNA from the mother. So we have extranuclear inheritance, so DNA that's not found in the nucleus can be passed down in different ways. The next one is X inactivation. This is only found in females because we have 2 X chromosomes. Males only have 1, so whatever they get on their X, that's what is expressed in the male. The X inactivation is a type of inheritance called epigenetic inheritance. This is when you have modification that can occur on a single gene, or you can modify a whole chromosome. And this modification that happens on the gene or chromosome, it can happen during egg formation, so during meiosis in the female. It can happen during sperm formation, so during meiosis in the male parent. Or this can even happen in really early stages of embryo growth and development. And these modifications that occur at these different times to the genes or chromosomes is going to alter that gene expression. And this alteration of the gene expression, it's going to be fixed during that individual's lifetime. So if you modify a gene during egg formation, the individual that comes from that egg is going to express this modification throughout its life. If you modify a chromosome during embryo development, so while the embryo is growing and developing into the fetus, then that embryo, that individual that comes out of the embryo, it's going to have this altered gene expression for its whole lifetime. So X inactivation is a form of epigenetic inheritance. So one of your X chromosomes in the somatic cell of female males is inactivated. So remember, females have 2 X chromosomes. One of them is randomly turned off during development. The other one is going to be active, or it's going to be expressed. And we know that this occurs because there's 2 lines of evidence. So we find that in female cells, we find the structure called a bar body, and it's only found in female cells, but not in male cells. And this is specifically in cats. And then we also use this to explain why only females can have the calico coat color pattern that we see. You never find a male that is a calico. So what happens during this X inactivation is that we have a permanent inactivation of one of the X chromosomes in the female, and this is random in the cells. So sometimes one X can be turned off, other times the other X will be turned off in a cell. And this allows some cells to be different than other cells. The cell will form a patch in that cat's skin. So this is a X-linked gene for coat color. So we have an orange allele, so XO, and then there's also a black allele, or XB. So in order to get a calico cat, we have to have a heterozygous female that has XO-XB. So what happens is that in this individual cells, in this heterozygote individual, those cells will randomly turn off one of their X chromosomes. This allows half of the cells to express the orange allele, the other half of the cells will express the black allele. And then you end up having patches of skin that will be orange if they come from the orange cell. Other patches of skin will turn black and they come from this black cell. These female mammals that show this X inactivation for the X-linked gene, we call these mosaic animals if it's an animal different from just the calico cat. So female mammals, we see this a lot when they're heterozygous for these X-linked genes that code for fur color. We see X inactivation occur in females, and I just want to quickly go over why this X inactivation phenomenon occurs. And it's believed that this occurs due to something called dosage compensation. So this is where inactivation of one of those X chromosomes in the female, remember females have 2 X's, it gives equal levels of expression of any X-linked genes that may be on these X chromosomes. And this is because, remember, females have 2 X's. If we turn one of those X's off, randomly turn it off, then we just have expression of one of those X's. Since in males are XY, they already only have one X, so that's what is expressed. So this is dosage compensation. You'll have an equal amount of those genes on your X that are expressed because only one X is being active at a time. Another complex inheritance pattern that we're going to look at is genomic imprinting. This is another type of epistasis, and this is where a segment of DNA on a chromosome is imprinted or it's marked somehow, and this imprinted or marked segment of DNA, it can affect that gene expression. Remember, this is epistasis, so if we change this gene expression, it's going to occur throughout the individual's lifetime. So these imprinted genes that have been altered or marked in some way, they don't follow the Mendelian patterns of inheritance, the two laws where we have the law of segregation and the law of independent assortment. So this is why it's considered one of these complex inheritance patterns. So instead of following these Mendelian patterns that we've been practicing a lot in lecture and lab and in chapter 16, the offspring, if it involves genomic imprinting, these offspring are going to be distinguished between and expressed, or they distinguish between and express either their maternal allele or the parental allele. So either the allele they get from their mother or the allele they get from their father. But they don't express both of them because one of them is marked to be turned on, the other has been marked to be turned off. So we have these different complex inheritance patterns. So far we've talked about epistasis and continuous variation. We've talked about what can happen with gene linkage if genes are found on the same chromosome. We talked about genes that are found in other parts of a cell, not in the nucleus. Those are extranuclear inheritance. We talked about X inactivation and genomic imprinting, which are a type of way that we can either mark or turn on or turn off certain genes or certain chromosomes. The last complex inheritance pattern is the maternal effect. This is where only the mother determines what the phenotype of the offspring is going to be. So this goes into the maternal generation affects the offspring's phenotype. So the genotype of the father and the genotype of the offspring doesn't matter at all. It does not affect the offspring's phenotype. And we're going to look at an example here. This is a snail. And these snails, you either have individuals where the internal organs spiral to the left or they're going to spiral to the right. And this spiraling either to the left or to the right, so clockwise or counterclockwise, however you want to distinguish it, left, right, it all depends on the genotype of the mother. So these are maternal effects. So for example, if we have these two crosses up at the top, so we have a big D crossing with little d, and little d crossing with big D. So normally, using Mendelian genetics, all of the offspring will be big D, based on just looking at the alleles and how the gametes are formed, how fertilization happens. All of these offspring will be big D. And this is what we find. So all of the offspring have big D in their genotype. But we find that from these different crosses, in the cross on the left, all of the offspring are dextral or they spiral to the right. The cross on the right-hand side, all of the offspring are synstral or you have spiraling to the left. So they have the exact same genotype, big D, for all the offspring. However, some go to the right, some go to the left. And if you look at the parents, the mother matches up what the offspring are. So on the left cross, your mother is spiraling to the right, so then all her offspring spiral to the right. On the other cross on the right-hand side, the mother is spiraling to the left, so all the offspring are spiraling to the left. So the only thing that matters is the genotype of the mother. That's what's going to determine the phenotype of your offspring. So this doesn't make any sense. It doesn't match up with our Mendelian genetics. So this is where we have this maternal effect. And this occurs when the new individual is in just an egg form. So this inheritance pattern can be explained by egg maturation, what happens in females. So if the mother, she produces an egg, and the mother is big D, those big D products enter into the egg. That egg will produce that big D or right spiraling offspring. If the mother is big D, those big D products go into the egg. That will cause that offspring to spiral to the right. In order to get a left spiraling offspring, the mother has to be little d. Those little d products go into her egg. That causes the individual to develop spiraling to the left. So again, all that matters is the genotype of the mother will determine the phenotype of the offspring. The offspring's genotype doesn't matter, the father's genotype doesn't matter, just the mother's. So here we have our 7 different types of complex inheritance patterns. And basically, there's more to this chapter, like you can do examples of different types of genetic crosses that show gene linkage or show maternal effects. But we don't have enough time for that, so this is kind of just a quick overview of some more complex inheritance patterns and how genetics or inheritance isn't as simple as the way we started off with, where we just have dominant recessive traits. They follow Mendel's 2 laws. We can predict the offspring phenotype based on the offspring genotype and all that stuff. So genetics is very complex, and we're always learning new things about the way that we inherit genes, how they're actually expressed, how the environment influences that expression, and so on. So with that, good luck on the final. There won't be a lot of questions from Chapter 17 on the final. Really focus on doing monohybrid, dihybrid crosses, and really understanding that information. And then, of course, you'll need Chapter 15, so the different types of cell division.